Thank you, Dave. I want to thank Dave Bradford and everybody else for inviting me out here. Hart Welch, Don Ashton, Jamie Patesco, and Carrie Schlick. And of course, Priya Nanjapa Mitchell and Ernie Garcia of Park, who really made arrangements for my 16-hour flight from South Carolina. Thanks for that. I could be in Australia right now. But anyway, I'm going to talk about long-term studies, amphibians, and let me see if I can make this work. Oh my gosh, what? Nope, let's see. That seems to be going backwards. Don, what do I do?
Yeah, one direction's backwards, the other direction should be forward.
It works the same way back east. But it doesn't seem to work at all here. Let's see. It's funny, it worked a minute ago. Oh, there you go. I don't know what I did, but anyway, but two, as David mentioned, there's DAPTF and there's Park. And I want to say a word about those first. And, oh, man. Wait a minute. I found another button, and it works. All right. Okay, let me show you what the difference between these two groups is. Not much. They both deal with amphibians, and they're groups of people who are interested in amphibians. Everybody in this room is. You wouldn't be here. One slight difference in Park and DAPTF is that Park also deals with reptiles because reptiles have the same, and their problems are as great as those of amphibians. Fortunately, amphibian biologists have really done a tremendous job of identifying the problem and trying to do something about it. We're just now at the stage with reptiles where amphibians biologists were maybe 10 years ago. But in either case, both groups of animals have major conservation problems. Hey, Don, can you hear me? Let me just do this or something. Next slide. Okay, I'm not going to talk about reptiles. I could, of course, as I just said, because they have their problems too. But if I just do that work? Okay, I'm going to talk about amphibians. And I'm not going to talk about these groups of people, DAPTF and Park, because the point is amphibians. That's why we're here. And for instance, back east, there's the gopher frog. Under certain political climates, it would probably be an endangered species because we see so few a year. And next is, for instance, you have the red salamanders, which seem to be very common. And this is one thing Park is really interested in is let's keep the common species common. Let's identify the rare ones and the problem ones and go from there. So here's my objectives today. First is to show you some amphibian studies based on what we think of as long-term studies that show you some interesting things that we didn't expect, and I don't think anybody did. Give you what we consider a template for doing long-term research. And last is identify what we've learned from it. And by this, I don't mean what we've learned about amphibians, but what we've learned about the importance of doing long-term research and how DAPTF and Park attitudes and groups can help. Because one of the most important things is not just doing the science, but then taking that science and making sure that not only other scientists understand it, but that the public and politicians understand it too, which is a lot harder than making the scientists understand it. But that's what we have to do. Next. I'm from an area of the country where you can see this is a number of species that are that the darker it is, the more species of this is reptiles and amphibians, biodiversity. Next. Next slide. And it's what I consider, I call these, they are hidden biodiversity because they're hidden not only from us as scientists, but but just the general public. They're hidden from view. Bird people have done such a good job of making everybody care about birds because you can see them all the time. Well, our animals just don't see them all the time. Yet, go ahead, next. For instance, you have a place like this in southern Georgia or South Carolina or Alabama, just a pine forest next to a wetland. And in that forest, right there, and people don't even know about it, you have, next slide, all of this requires that habitat. Even though some of them terrestrial, some of them aquatic, they all require this habitat, and they're all right there. Next slide. Hey, another thing, why study amphibians? Well, DAPTF and other groups have made it very apparent we got a big amphibian problem. Next slide. The salamander diversity back east is a good reason to take that group to study, but also it's a wonderful group to get the general public interested, especially if you have live animals and show them. Next slide. For instance, just in Georgia alone, have 50 species of amphibians, and a great number of them are, 50 species of salamanders, I'm sorry, in Georgia alone. And they're all different. It's wonderful to be able to study those, but it's also a great group to make people aware of, here's a group of animals we want to keep around. Next. Next. That spot right there is where the Savannah River Ecology Laboratory is. It's right in the Midlands of South Carolina and Georgia. Next species, next slide, or species. But right there are 51 species of salamanders in that particular area of South Carolina and Georgia. And they breed in different ways. Dee Dee mentioned, for instance, the animals breeding in the different aquatic habitats. Some require permanent waters. Some require seasonal wetlands. A few are terrestrial in that area. You have more terrestrial ones out here, but and then there's some that breed in both permanent and seasonal. Next slide is, okay, who are we and why? Next. University of Georgia. I'm with the University of Georgia. I just live in South Carolina. Next slide. And right on the Savannah River Ecology Lab is located right here. Next. And along the border, this is Savannah River site over here. This is in South Carolina. This is Georgia over here. And this is the Savannah River. So this is one reason for the high diversity of species in this area. We have so many different habitats. Next situation. But here's the irony, the paradox. Okay? The reason, next slide, Savannah River site is there is because of 5 nuclear production reactors. And just a quick note, if you wonder where all the most protected lands in this country are, it's the military bases. You don't want them closing military bases. You know why? Because people can't get there and build malls and highways and other things. They blow up a few things, but the animals are fine because protected habitat. Next slide. This, for instance, is a satellite view of the Savannah River site. It's the largest area of fenced-in land in the eastern United States. 300 square miles. That's a lot of land back there. And the reason it shows up green is because that's the way the upper coastal plain is supposed to look. This is the development, agricultural, other types of impacts. This is the Savannah River site. Next slide. If you just look at wetland habitats, which we're going to focus on for the rest here mainly, but the amphibians do require the terrestrial uplands, but there are many different kinds of wetland habitats. Next slide. And next slide. But the reason they're there, the reason they're protected is because you can't get there, right? Because it's a protected site. 300 square miles of mostly protected. Now we have to put some controls and caps on the Forest Service and other groups that try to help out, but for the most part, it's an ecological preserve. Next slide. And one of the things is a group of wetland types called Carolina bays. You'll see this term come up, and I kind of use it sort of without thinking about that most people aren't familiar with what a Carolina bay is. But it's the seasonal wetlands. This is a small one. This is a large one. They come in all sizes, but they're nearly always egg-shaped, and they're oriented northwest to southeast. Northwest, southeast orientation. And there's 250,000 of them across the Atlantic coastal plain. Origin is not quite agreed on. Next slide. One of the things I'm sure a lot of you here are familiar with is drift fences. I'm going to mention drift fences. It's just a technique. I'll mention it, and I just want to pass through it real quick. But in short, next slide, is what we do is put pairs of buckets or traps of some sort on either side of this fence. Next slide. Put them about 10 meters apart, and then check the fence every day. Animals hit the fence, go one right or left, and fall in the bucket. It works great for salamanders and for many kinds of frogs. Next slide. Okay, Savannah River site has more species of reptiles and amphibians than any other government land area, including the Everglades or the Smokies, any other in the United States. And the studies have gone on for a long time, starting back. I think this may have been the first. I mean, I wasn't there, but this may have been the first herpetofaunal survey done. Was back in the 1950s for a government project. Anyway, and you can see over the years now that we know of have more than 100 species on the site, and it's funny, it still hasn't leveled off. We still get new ones. Next slide. And I need to give credit for a lot of people have been involved. Many students, many visiting professors from different faculty from different places. Just all kinds of people, and I didn't want to give an impression that, I'd just gone out and collected the data I'm fixing to show you. Next. And it is the long-term study at Rainbow Bay. That's one of those Carolina bays. Particular one. Next slide. And I need to mention particularly the people who were involved in that, because these people started, Ray Simlich particularly, kept this project going. And next slide. And David Scott, who is still there, checks these traps every day. Go ahead. Next slide. Okay. It's been going for 26 years, this research at this site. Next slide. And Rainbow Bay, we've checked the traps every single day since, and this includes all the holidays, since September 1978. Next slide. There's 26 amphibian species. And, this Carolina Bay, by the way, is no bigger than this auditorium. It's about the size of it. Very small. Next slide. Capture about 24,000 a year. Many of the species are focused on for marking. Next. Next slide. And one of the things which has been revealed to us, there's several of these species, I think now 7 or 8, have recaptures because they're individually marked after 10 years. I mean, they live a long time. We did not expect this for a little mole salamander. David Scott and I the other day caught one that had been toe clipped, and we knew, it was no question about it because it had been caught before. It was 13 years old. And this is a little bitty gray, ugly salamander. It's really something. Next slide. And this is the real measure we have, of course. This is what the Department of Energy, who supports us, a lot of us understands more than publications in Science and Nature. Again, this is much more important to the importance of our study. But that's a sign we keep out at Rainbow Bay to when we're showing groups around out there. Next slide. Quick thing of the process. I know you all know how amphibians work, but for this particular, the ones I'm talking about that breed in these seasonal wetlands, they of course lay their eggs. They go through a larval stage. Very complex life cycle. They emerge here as juveniles, and then some of them actually become pedomorphic and stay. Tiger salamanders do out in the west, for instance. Stay pedomorphic but generally they go on the land and then go back through the cycle. Next. So anywhere you break that cycle, you're helping with amphibian decline. These particular wetlands have a particular cycle. They go through, it's fairly predictable for seasonal aspect. It's likely to dry up in the summer. It's likely to have water in the winter. But not every winter, not every summer do you have the same phenomenon. It varies. Next. And so an important thing is what we call a hydro period, which is just for simple purposes the number of days the wetland holds water. And I think Hart mentioned this, and Dee Dee too. I know Hart did mention fish just don't work very well with most of these kinds of amphibians. But fish are native, they're natural, but they're not natural in a seasonal wetland that's going to dry up at least every 2 or 3 years. And then so you have some kind of poor investment sometime by the animals, and it dries up before, whether they're fish or not, dries up before they can make it. So it's a dynamic, exciting place to be, an amphibian, but you don't know if you're going to survive or not. Next. Okay, I want to go through a little, and I just picked, this is just a poster child kind of picture of spadefoot toads. You're all familiar with, heard of spadefoot toads. And this is longer than a master's thesis. This is at Rainbow Bay. These, on this axis, are going to be how many juveniles were produced at Rainbow Bay. Remember, we have this encircled with a drift fence. So we catch everything coming in and out of that place. And for 26 years now. Okay, so this is also a natural habitat wetland that's not affected by anthropogenic human influences. It's just it's about as natural as you can get these days. Okay, next slide. Oh, I forgot. I was just going to show you to scale. These bars here now are kind of reduced because of this. Because of this, rather. Okay, next slide. Same thing. Goes 10 more years. There's none. They've gone. There's no more spadefoot toads going to be produced after 21 years. We've finally shown they're not there. Then, of course, we go 2 more years. Next slide. Ta-da. And that year, 5,000 were produced. So, and then, of course, next slide. We've got this situation of these 2 big years. And next slide. And we want to look at, okay, what about the females coming in? I hadn't been showing you that, but this next slide shows you in the year that so many were produced here, this is how many females. About 400 females came in. Next slide. But here was another year back in the 90s that many females came in and there were none produced. Next slide. Next slide. And then the year this bunch with 3 animals. That's it. 3 females, gravid females came in, and that was the outcome. Here, 400 come in, and that's the outcome. Wow, it just shows you there's so much yet to learn about these animals. Next slide. So, we've got this situation, these different dramatic events happening. Next slide. And, okay, and I've put these now into scale because, next slide, these are those big years. Remember, 3,000, 5,000. Next slide. This is what happened 2 years ago. Next slide. That's how many juveniles came out of Rainbow Bay, a little place like this. That's going to keep them for a long time. But what is really keep that population alive for a long time? But here's an interesting thing. You think about it, and as we were discovering this, look how long these females have to live, because the ones here, this is the only wetland right there they can be producing in. Any that came out here or here had to have come over an almost 10-year period. So females have to live out, so they have to live out in that terrestrial habitat. It's got to be protected too. You can't just protect the water. Next slide. Just showing you that the same phenomenon works with these other species. Look at the variability in, this is Ambystoma talportium, and the number of breeding females coming in over different years. Next slide. And the sporadic, intermittent, the unpredictable, haphazard development of juveniles. Next slide. Same thing with marble salamanders, Ambystoma opacum. That females are much more commonly coming in there these days. Next slide. And yet they had one of these big boom years, but it was different from the Scaphiopas. You notice, I mean, the spadefoot toads were over here. Their big marble salamanders' big year was about 8 years ago. Next slide. And so, what if you'd done your 5-year study on Ambystoma opacum about their making conservation decisions and all based on this 5-year period? Next slide. Compared to, say, this 5-year period. Be completely different. And I mean, and I think I like one of the things Dee Dee was saying is how they're finding as they look at different, go to different forest situations, you find differences. It happens. That's the way there's still so much to be learned. Okay, next slide. Okay, I'm going to quickly run through this, just show you some general principles determined about hydroperiod. Next slide. This is based on a paper published by Joe Peschman and David and Ray and I, next slide. And I guess you can't see that, but this is at the bottom is the hydroperiod. This is the number of days the water was in the Carolina Bay, in the Rainbow Bay. Next slide. If you look at the number of species that bred, the more water, the longer the hydroperiod, the more species actually bred or came in that year. Doesn't mean they were successful, but they came in that year. Next slide. If you look at the number of individual amphibians, the more water, the longer the water, the more likely you are to have, and this like gets up to 75,000. Next slide. Taking a particular species, this is just taking mole salamanders, there's a threshold. Every species has that, and the problem is this is a conglomeration of those thresholds, but it has a threshold. In other words, you can model this and determine how long does it have to, how often would if you take the age, how often would you have to have water at a certain level for this species to persist. And then the next slide, and in that way this is the most interesting one of all because there is no relationship that's very detectable. These are Rainbow Bay animals from this species, mole salamanders, females coming in, breeding. Doesn't have anything to do with the hydroperiod. It's really hard to know what's happening. Next slide. But I point out, each one of these points here is a year. Okay? That's how much, to get these kind of patterns, you have to do this for a year for each point. Next slide. And just again to show you, I just picked sample species before. Here's a tiger salamander, which is a western species. This was the last time they produced successfully at Rainbow Bay. Are they gone? Do we tell Department of Energy, who owns that property, that Okay, it isn't an endangered species here, but it is in parts of California. And here you've just eliminated them at this place. And next slide. Another species is the ornate chorus frog. Same thing. Haven't seen one, or haven't successfully produced one since, gosh, it's back in the late 80s or early 90s. They gone? I picked these two species for a reason, because now I want to finish up with just showing you one other quick study, which was we were just fortunate to be able to do. Next slide. Okay, Ellington Bay is another Carolina bay. It's about 10 miles away from Rainbow Bay on the Savannah River site, and for there studies have been going on for 38 years. Starting with studying turtles. Next slide. So I was glad Hart had a turtle in his pictures too. Okay, go ahead, next slide. It's one of the Carolina bays. Next slide. And there's about 60 species at this site of reptiles and amphibians. Next slide. It's been surrounded by a drift fence. We put in the first drift fence in, I think it was 1968. And it hasn't been run continuously like Rainbow Bay, but it's sort of intermittent. But for several year periods at a time, at least for over 10 different years for the full year. Next slide. I talked about the water level and the seasonal fluctuation. Ellington Bay can look like this in 1 year, and 2 years later, that's a meter deep, and that's about a meter and a half deep. There's a gator hole right out here that's 2 meters deep, but right here it's a meter deep. Next slide. It can look like this a year or 2 years later in the summertime, and on through the winter. Next slide. The drift fence I mentioned. Go ahead. Next slide. Set up the pitfall traps. This fence next slide, is a mile long, and we use different kind of traps this particular year I'm going to talk about. Go ahead, next slide. And for the 1 year period, all amphibians that were moving in or out, we determined their stage and sex and weight to get a weight measurement. Next slide. Okay, next. All right, water level. Next slide. Over the course of time, the water level at this particular Carolina Bay has fluctuated like this, from about a meter deep to drought periods where there is no water. I mean, you can walk out there and not get your shoes wet. Drought periods can last for, like this one over here. Next slide. There's water under the surface, but it's not breeding purposes for an amphibian. It's useless. Okay, but this drought was a period here for about almost 3 years. Next slide. And so we set out to ask this question, next slide, of how productive can it be and how important is the land, next slide, to these animals. Just to give you a little more background on Ellington Bay, Rainbow Bay, by the way, is as I said, about 10 miles over here. This wetland, this was 50 years ago, an aerial photograph before the Savannah River site was taken over by the Department of Energy, and it would be a degraded wetland area at that time. If it persisted, there probably wouldn't be any. Well, there'd be probably a few amphibians there, but not many. Next slide. And now, though, it's kind of forested around it because it's been set aside. It was set aside by Eugene Odom 50 years ago to protect this and many other sites on the Savannah River site. Next slide. Questions we had. Slide. I'm not going to address all of these, but we set out about with these kinds of questions that we were asking. Next slide. Next. And the reason we did it is I had 6 new graduate students that didn't know anything about amphibians or reptiles, and I want them to learn them. I want them to learn how to set up a drift fence, how to run a drift fence, collect data. This was back in 2002. And so we did that. We set it up. This fence had been run for a third of a century anyway, off and on. But it had been down for 3 years because of this drought. We weren't getting anything. So next slide. So we'd taken the fence down. And I think I've already shown you this. Go ahead, next. And then the drought thing. Next slide. This is the area we're talking about. The students came right here. We started putting in the drift fence. Next slide. Here, and we finished it, got it completely encircled on February 1, 2003. And then next slide, you're supposed to look at that raindrop. Next slide, this is what happened at Ellington Bay, starting on February the 4th. We put that fence in, been a drought. I said, well, you know, we might go a year and only catch just a few racers and rat snakes and, you know, bufo, but well, we ran that thing every single day for at least once a day for a year. And this, we sort of kept a record. These are initials of people. That's me right here. How many times you spent? Chris Winnie gets his picture there because he got the record for running it the most times. These people hardly got put on the publication. Next slide. So we checked these every day for 1 year. Next slide. This is how many amphibian species we got, how many reptile species, 24 species in that 1 year. Next slide. Got 1,100 individual captures of reptiles. And how many amphibians do you think were? Now remember, it had been a drought. How many amphibians do you think we got? Next slide. That's how many amphibians we caught in that 1 year. And this was great. I mean, those students know a lot about 24 species of amphibians. Next slide. Just to give you an idea of some of them, we caught 232,000 leopard frogs. And this is Luke releasing some. Of course we catch them and release them on the outside if they were going out and vice versa. Next, 100,000 bufo. There's a few reptiles mixed in here. 3,000 mole salamanders. Keep going. Some were common. Next slide. And some were got your attention more than others because there were over 100 cottonmouths that came in. And this place had never, this is one of the long-term interesting things. Ellington Bay had never had cottonmouths before. They just knew that there was a lot to eat there. How they know? They came all the way from the river, a mile away, overland. And the ornate chorus frog, that's one of the ones, remember, that was missing from Rainbow Bay. Okay, it's doing well at Ellington Bay. Next slide. And then there are some rare ones, like the gopher frog. We only got 3. Next slide. And some of even this is the dwarf salamander. Now it turns out it has been missing from Rainbow Bay, and that year it can't return to Rainbow Bay, so it'll probably recover. Next slide. Okay, this is an important slide because it shows you this is biomass. I said we didn't weigh every frog, of course, but we weighed 100 leopard frog metamorphs and found out how much that averaged and then calculated the biomass. So leopard frogs were the primary animal in terms of biomass. Bufo were, the toads were next. Mole salamanders, next slide. These are the 2 species that, remember, had not shown up at Rainbow Bay for 8 or 10 years. And they were, that would be 50 pounds of tiger salamanders. But the total biomass, putting it in English, is, next slide, 3 tons of metamorphosing amphibians came out of Ellington Bay. Just incredible. And we sometimes ran that bay 24 hours. You would go run the fence. I remember running it one night from like 11 till 4 in the morning and catching animals in the buckets. You'd get around to the end of the mile, start again because their buckets were full again of like Rena coming out. And then by then the new crew, the 4 o'clock crew had arrived, and then they took over. So there was a lot of effort put into this. Next slide. But it's worth it, because amphibians are worth it. They're an exciting group of animals, and there's so much to be found out about them. Okay, next slide. And just to end up here, what did we learn? I told you I would tell you about, I've shown you kind of what we learned about amphibians, but what did we learn from long-term studies about conservation, about how to live our lives, and other things? Next slide is one. Next. And then I'm going to tell you exactly what I mean by each of these. Next slide. You've got to be cautious. The reason we have to be cautious, next, is we don't know a lot. I mean, we're talking about a study where we thought we knew all about spadefoot toads or tiger salamanders. We don't know anything. I mean, yeah, we know something, but we know enough to make it clear that we've got more to learn. Next. And so we don't want to, for instance, go tell a government agency, oh, you got to protect this wetland because, or you don't need to protect this area because something's gone when it's really not, or that you have to protect it because it's so rare, and then you get 50,000 of them one year. That's not going to be very credible. We just have to be cautious about how we do it. The patterns, we don't know all the patterns, next, and the variability. And it's true here, it's true everywhere that these animals occur. We just have to, we know this already, but we just have to be careful, and we have to convince our public audiences and the politicians, as I said, that, you know, this is reality. This is the way it is. The variability is something that's real in itself. Next. The challenge, next slide, is funding for research. You all know that. I think I don't have to say any more about it. But we need to be supportive. Those of you in federal agencies or state agencies need to be getting the funding to the people who can do the research. The research is going to tell us, next, we need to protect habitats. And as I said, the irony of the federal protection, forest lands, or U.S. Forest Service has got tremendous opportunities to protect habitat. And just how they do it and is what's important. And I cannot say enough about how important it is that we educate not just each other at a meeting like this, but that we go out and educate school children, the news media, politicians, if they'll listen, and we need to be telling everybody about how exciting these things are, how we need that we're never going to know completely about them, but if we lose them, obviously we're not going to know anything. Next. And the optimism part is, next slide, is there is hope that once again, before too long, we will be using science-based decision making. All right? I think it's what, 731 days? No, I'm not. I don't know, but it's got to be soon. And we know this, and we have to get there. Next slide. We've got to continue amphibian research. I mean, we got to do it everywhere. I know Gary Fellers has a great program out here. Dee Dee's talked about her program. We have plenty of things back east that in the southeast, particularly the northeast too, that lots of opportunities and things that need to be supported, continued, and then the really important thing, last slide, is any study is a long-term study. You just have to keep doing it, and you have to support people who are doing them. And if it hadn't been for the Department of Energy, the irony there is the Department of Energy you don't think of as a research, an ecological research group to support ecological research. But that's who supported all those Rainbow Bay studies for more than a quarter of a century now. Anyway, I appreciate your letting me talk to you. And remember that amphibians are very important for one other reason. Next slide. That is, they serve as a food base for the really important reptiles, okay? All right, thank you very much.